This protocol can capture real-time kinetics on a single cell level. We can measure individual cell parameters in response to HIV infection and associate early signaling events with delayed steps of the viral life cycle.
This is an integrated, scalable alternative to traditional imaging methods using a novel optofluidic platform for single-cell sorting, culturing, imaging, and software automation.
This is the first established method for nanofluidic, high-throughput, longitudinal single-cell culture and imaging. This technique can be broadly adapted to study cellular signaling kinetics and dynamic molecular interactions in a variety of disease states.
Visual demonstration of this method is important to convey how the experiment is set up, how cells are optically sorted into pens, and how to set up infusions through the chip.
To begin, prepare fresh fluo4-AM loading solution by adding 25 microliters of 100x concentrated detergent solvent and 2.5 microliters of 1000x fluo4-AM to a 1.5 milliliter tube, then vortex to mix. Pipette 2.5 milliliters of culture media into the loading solution and invert to mix. Centrifuge 2 million MT4 cells at 500 times G for 3 minutes. Then remove media and resuspend the pellet in 2 milliliters of the prepared fluo4-AM loading solution. Pipette resuspended cells into a 35 millimeter Petri dish. Incubate at 37 degrees Celsius for 15 to 30 minutes. Then incubate for an additional 15 to 30 minutes at room temperature.